In this video, we'll begin our look at bacteriophages. Remember, those are the viruses that infect bacterial cells. There are three general types of bacteriophages based upon ultimately the relationship that each one has with its host cell. There are lytic or virulent phages, temperate phages, and also filamentous phages. We won't be discussing the filamentous phages, and in this recording actually we'll only look at the lytic phages. We'll discuss the temperate phages in a separate video. There are two general strategies for phage replication. Replication is the ability of a virus to make more virus. You can either have a productive infection, in which new viral particles, new virions, are actively formed and produced, or you can have a latent state or latent infection, in which the viral genome remains silent within the cell, but each time the cell divides, the viral genome is replicated and then passed on to the progeny. So those are the two general strategies for phage replication. If we take a look at this diagram here, which comes from your text, you can see the virion, which would infect the host cell. And then you have your two different strategies for replication. In a productive infection, once again, more virus would be produced. Up underneath the concept of a productive infection, you can either have a lytic infection, in which the release of the virions causes the host cell to lyse. This would ultimately lead to the death of the host cell, or in some cases, those new virions can be released in a manner that does not cause lysis to the cell. In many cases, this is referred to as budding, and when the virions bud away, they usually take a piece of the cell membrane, becoming enveloped viruses. But that's not 100% always going to be the case. In any regard, when virion release does not lead to lysis, the host cell can continue to thrive and live, and therefore continuously release virion. But in either case, you can see more viruses produced, so both of these infectious types would be considered a productive infection. In a latent infection, or the latent state, you would not actually see any new viruses produced immediately. Notice, ultimately the virus introduces its genetic information into the host cell, and then ultimately that genetic information is accepted and taken up by that host cell, typically incorporating the viral genome into the host cell's genome. Ultimately, as the host cell reproduces and causes the formation of new host cells, that particular viral genome is copied and carried forward with it. So at some point in the future, typically, we would see that viral genome began to be expressed, and at that point it could become a productive infection. But with the latent infections, they initially start out not being a productive infection and can ultimately be triggered through a variety of different means to become a productive infection much later on. So we want to take a look at lytic phage infections here, and we'll begin to use the T4 phage, which I mentioned to you in the previous lecture, as a model. As I said to you before, a lytic infection is a productive infection, so we will actively see new virions produced. Once again, the T4 phage, which infects E. coli, is typically the model that we use, and you can see how quickly this process takes. It's about 30 minutes long. You'll definitely need to keep in mind the 5 different steps involved in the lytic phage cycle. Ultimately, we go through 5 steps of attachment, genome entry, synthesis, assembly, and release. These are not that complicated. To be quite honest, the names basically indicate exactly what's going on or exactly what's happening. So we began by taking a look at attachment. With attachment, you are actually going to see the T4 phage attach to the cell wall of that particular bacterium that it's going to infect. As you see here in the top right hand corner, those tail fibers contain spikes, which we mentioned before, that will help it attach to the cell wall. Typically, the attachment is to a molecule that's integral to the particular organism. In other words, it's not an arbitrary molecule that may or may not be there. Typically, these viruses attach to molecules in the cell wall that absolutely must be there. It just increases their ability, or increases the chances rather, that they can find a host cell. because if a host cell happens to lack that specific molecular component or receptor, then, well, the host cell will become resistant to infection by that phage, and that would not favor the continued existence of that particular phage. So the attachment step usually involves, once again, the spikes located on the tail fibers. Keep in mind that they will attach or bind to some critical component within the cell wall or membrane of that particular bacterium, therefore increasing the odds of being able to cause infection. Next up, we have genome entry. This is when the viral genome is introduced into that particular bacterial cell. In order to enter the bacterial cell, you must degrade or break down the cell wall. So the T4 phage actually has T4 lysosome. The T4 lysosome will degrade the cell wall, and then ultimately the tail unit contracts. And when it contracts, it almost looks as if the T4 phage is squatting or sitting down on the cell wall. But as the tail unit contracts, it will inject its viral genome into the bacterium and ultimately allow for entry. Notice the capsid remains outside. The capsid never enters. It's just more of a protective coat that is protective until the viral genome can enter the particular host cell. Next, we have synthesis. Once that viral genome enters the bacterium, that is the host cell, that is, ultimately it begins to promotes the transcription of viral genome, and the production of phage proteins. The phage DNA is replicated, and as you can see here in purple, the host cell's DNA, the bacterial cell, is actually going to be degraded. This decreases the chances that the host cell begins to try to continue to make host cell proteins, and since the viral genome is going to be dominant, well, ultimately that increases the odds of you actually producing more viral proteins. Now, the viral proteins are divided up into two major categories. You have early and also late proteins. The early proteins, as it says here, are translated within minutes. Those are the proteins typically aimed at trying to take over or hijack the host cell. There's a nuclease that actually degrades the host DNA, and as that host cell's DNA is degraded, once again, that would inhibit the ability of the host cell to produce host cell proteins, because without the genetic information being intact, well, the host cell just couldn't make it. So as you degrade the host cell DNA and along with some of the other earlier proteins, you ultimately hijack that cell and direct the synthesis or production of viral proteins. Most of the viral proteins that are produced later on are actually more the structural proteins, the capsomers that help make up the capsid, and also its tail. So those don't come along until the phage is actually just about ready to move to the next phase or next stage of the infection cycle, which is assembly. With assembly, we're going to put together all of the various components. Most of these components are going to spontaneously come together. We say that they self-assemble. For instance, many times the capsid just automatically comes together. So those capsomers, once they're produced, just unite automatically. You can see other structures such as the base plate, tail fibers, the tail as well. They all have to come together. But before that actually occurs, you actually have to incorporate the genetic information into the capsid in order to form the nucleocapsid. Now, in some cases, the assembly of these particular items requires help. So in some cases, proteins actually function to kind of align everything and make sure that they're going to structure themselves correctly. But as I said to you before, many other components self-assemble. It's a very unique aspect that is, to be quite honest with you, is studied extensively because it is thought to be very useful from a molecular standpoint. As you cause the assembly of more and more virion, what begins to happen is that the cell becomes full of virion, and without the ability to expand, ultimately what begins to happen is that lysozyme is produced on the inside of the cell. In this case, the lysozyme, as it's produced on the inside, will degrade the cell wall, and once the cell wall is degraded significantly, you will see that the cell lyses. Once the cell lyses, it's now going to be free to release the virion that was produced during that active or productive infection. Typically, The burst size, which is the number of organisms released once lysis occurs, of a T4 phage is going to be approximately 200 virions. And once again, this entire process would take roughly about 30 minutes for a T4 phage. And that is the overview of the lytic infection cycle.